effects of concentration on the enzyme functions. A good example of this is, imagine if you have a team of workers assembling toys. The more workers you have, the faster the toys will be assembled, up to a point. Once you have enough workers, adding more won't make the toys get assembled any faster because everyone is already busy working. Enzymes are like these workers. They speed up chemical reactions in our body. The more enzymes you have, the faster the reaction will happen, up to a certain point. Once all enzymes are busy working, adding more won't make the reaction any faster. This is why enzyme concentration is an important factor in how our bodies work. If we don't have enough of a certain enzymes, our body may not be able to perform important chemical reactions. And as we have here on figure 2, we go over that, and on figure 1 we show the reaction, and it explains in our method, in our introduction, and the conclusion we came up with. The hypothesis was that increasing enzymes concentration would increase the rate of the reaction. The prediction was that the rate of the reaction would increase linearly with the increasing enzyme concentration. Also, enzymes concentration is a factor that affects the rate of enzymes catalyzed reaction. The importance of this finding in understanding biology processes. Some of the limitations of the experiment, such as the need of more precise measurements. Some suggested ways that the experiment could be improved, such as a wider range of enzyme concentrations. As for the methods, the experimental setup for this experiment is as follows. 4 reaction tubes are prepared, each containing a consistent volume of distilled water. Varying concentrations of the catalyst enzyme are added to each tube, except for the 4th tube, which serves as the controller. A fixed volume of hydrogen peroxide substrate is added to each tube. The tubes are incubated at a consistent temperature. The volume of bubbles produced in each tube is measured over time. In the result text box, we have when the concentration of the catalyst in the reaction is increased, the reaction volume typically increases proportionally. This is due to the catalyst enzyme ability to improve hydrogen peroxide breakdown into a water oxygen. As a catalyst concentration increases, more enzymes molecules are available to aid in the decomposition of the hydrogen peroxide, causing the overall reaction rate to accelerate. And as for limitations and assumptions, enzymes are sensitive to environmental factors such as temperature, pH, and the presence of inhibitors. If the enzyme was exposed to unfavorable conditions, it could denaturalize, losing its catalytic activity, also known as enzyme denaturation. Also, finally, for our conclusions, our investigations into the factors affecting enzymes activity revealed the profound influence over environmental conditions and experimental techniques on the performance of these biological catalysts. We observed the enzyme's denaturation, substrate purity, mixing efficiency, equipment malfunction, and human errors can all significantly alter enzyme activity, potentially leading to erroneous interpretations of experimental results. And with this, as we saw in the lab, we messed around with it, and you can see if you add too much of something, too much or too little of something, the results always vary. So it is very important to always be consistent and try to have a controlled environment as much as possible.